Yes. So two different types of fermentation processes. There was homolactic fermentation, and depending on the organism, there was what else? Alcohol fermentation. And again, both of those guys were taking pyruvate, and they were essentially reoxidizing the reducing equivalents, NADH. Okay, so now we're going to just postulate what would happen if we had some sort of nice, strong oxidant around. Okay, so some sort of nice, strong terminal electron acceptor. So when that is the case, we have this nice, beautiful little oxidant around. We're not going to just go through fermentation. Instead, pyruvate is going to take a different pathway. Pyruvate is instead going to go through something called the citric acid cycle. We're going to talk ad nauseum about the citric acid cycle in this chapter. There's a couple of reasons why this would be the case, right? So number one, pyruvate, as is still really reduced, meaning that there's a lot of electrons in pyruvate that can still be harvested for the purposes of energy generation. That is the purpose of the citric acid cycle. It is going to take pyruvate and is going to completely oxidize it to the most oxidized form of carbon, which is what? CO2. So CO2 is the most oxidized form of carbon. Still have lots of electrons to take from pyruvate before we actually get to the state of CO2. Okay, so who is the best oxidant in the world? Oxygen, right? So oxygen is the idealized terminal electron acceptor. We didn't figure that out metabolically until the rise of oxygen in the atmosphere. So there are other possible terminal electron acceptors, right? So if you've taken, what is the microbiology course? Microbial universe. You guys have talked about all the various ways that microorganisms can eke out a living, right? So we can utilize things like nitrate, things like sulfate, as well as oxidized metals, just to essentially act as these terminal electron acceptors. It doesn't just have to be oxygen. So there are many other ways by which other organisms, not us, can utilize various things that they find in their environment as a terminal electron acceptor. Okay, so a couple more things. The citric acid cycle is considered a hub of cellular metabolism, right? We have been looking at this in the perspective of taking sugars, taking carbohydrates, and generating energy from them. But the citric acid cycle is not just limited to carbohydrates, right? Other various types of metabolites can enter the citric acid cycle. This includes metabolites from the degradation of fatty acids, as well as amino acids, right? So even though we consider this in terms of carbohydrate metabolism, other things can enter and other things can be utilized in generating energy with respect to the citric acid cycle. That is why it is considered a hub of cellular metabolism. Okay, so great. Now, what is overall the idea behind the citric acid cycle? What is the overall reaction that we see? So the citric acid cycle, and we are going to look at it in the form of carbohydrate metabolism. So we're going to start with pyruvate up here. But the citric acid cycle really starts with acetyl-CoA. Okay? So everything else, including amino acid metabolism and fatty acid metabolism, will enter at this point with respect to acetyl-CoA. So really, a citric acid cycle starts with acetyl-CoA. So it is going to consist of 8 separate reactions. So 8 reactions that are going to take that acetyl group on acetyl-CoA and completely oxidize it to CO2. In the process, we're going to take those electrons and we're going to reduce our various reducing equivalents. This includes NADH, or NAD+, as well as FAD. So we're going to produce NADH and FADH2 in the process. Okay, so another thing to note, it is a cyclic cycle. It is not just a nice little pathway that we have for glycolysis. So part of this is going to be the actual reactions that we're interested in, and then the other half of it is going to be regenerating the things to start it again. So the first 5 reactions are what we're actually looking at, and then the last 2 or 3 are actually just getting it back to the starting place, which is oxaloacetate. And then long term, the carbons that we put in on the initial cycle are not going to be the carbons that come out on the first round of the cycle, right? So again, we put in the acetyl group and acetyl-CoA. These CO2s that come off in this decarboxylation reactions, these are not going to be the same carbons that we put in the acetyl-CoA, right? Those carbons are going to come off in subsequent cycles, and I'll try to illustrate that when we actually go through the reactions. Okay, so long term, what are we going to get out of this? We're going to take our 2-carbon acetyl group. We're going to get 2 CO2s out of it. Most importantly, though, we're going to get 4 pairs of electrons. So 4 pairs of electrons are going to come out of that. 3 of these are going to be used to reduce NAD+ to NADH. So 3 are going to go to the formation of NADH, whereas 1 is going to be used to reduce FAD to FADH2. So we have 2 different forms of reducing equivalents that are generated via the citric acid cycle. And overall, if you want to look at the reaction, this is the balanced chemical reaction for what occurs in the citric acid cycle. Also note that with these reducing equivalents, we also get the formation of GTP, which is an equivalent to ATP as well. Okay, so we're going to go and start this. Before we ever start talking about the reactions of the citric acid cycle, we first have to talk about how we get pyruvate, which is where we left with respect to glycolysis, into the form of acetyl-CoA. We haven't talked about that. So that is our first little pathway that we are going to discuss, taking pyruvate and turning it into acetyl-CoA. Okay, so this is what is catalyzed in this reaction. We have pyruvate, we are turning it into acetyl-CoA. This reaction is going to be catalyzed by an enzyme complex called pyruvate dehydrogenase. Okay, so there's a couple of things to note about this reaction before we talk about this enzyme complex. So number 1, pyruvate, how many carbons does it have? 3, right? So pyruvate is a 3-carbon sugar. When it turns into acetyl-CoA, how many carbons does an acetyl group have? Just 2, right? So this is our little acetyl group of acetyl-CoA. This is what's going to enter the citric acid cycle. So in this first reaction, or this first multi-step reaction, we're going to take a 3-carbon sugar and turn it into a 2-carbon sugar via decarboxylation. So one of our products is going to be CO2, because we've taken essentially a carboxyl group off. Just as a note, anytime we decarboxylate something, it is going to come along with the reduction of some sort of electron carrier. Okay, so if we decarboxylate things, we're going to take a CO2 away, but we're also going to reduce an NAD to an NADH. Okay, all right, so let's start to talk about this pyruvate dehydrogenase complex, because this is a beast of a complex. Okay, this is something that is called a multi-enzyme complex. Overall, I know it looks really simple, pyruvate going to acetyl-CoA. This multi-enzyme complex catalyzes 5 separate reactions. Okay, so taking pyruvate and turning it into acetyl-CoA is not just one simple step, it is 5 different reactions catalyzed by 3 different enzymes. All of those enzymes reside on this multi-enzyme complex, so they're all tied together. I'm going to demonstrate how they're all tied together. So there are a couple of advantages to making these various reactions occur on the same multi-enzyme complex. A couple of those, number 1, would be an increase in rate, right? So if we increase or we guarantee collision with the next enzyme, we're going to increase the overall rate of the reaction. Putting all of these enzymes on the same multi-enzyme complex will also help channel our products to the next enzyme active site that they need to go to, right? So that's going to help in preventing any type of unwanted side products. And then, as we have demonstrated, a little bit too much, we can also control these enzymes very easily, right? So these are all going to be very easily coordinately controlled. Just as one more thing to note, we say acetyl-CoA, right? So we say coenzyme A looks great, right? Acetyl-CoA is actually this guy over here, okay? So the acetyl group that we're transferring and forming acetyl-CoA is up here. The rest of the coenzyme is this big, large thing, okay? But just note that the high energy bond that we are forming in this process is this thioester bond that occurs at the end of acetyl-CoA, where the coenzyme A is attached directly to the acetyl group. This bond, this nice high energy bond. Oh, one more thing. Oh, gosh darn it. Okay, so maybe I didn't mention this, and I probably didn't. Where does glycolysis occur in the cell? Cytoplasm, right? So glycolysis, unfortunately, occurs in the cytosol. It's in the cytosol. Where does the citric acid cycle occur? Mitochondria, right? So both citric acid cycle as well as electron transport chain are going to occur in the mitochondria, right? So before we can do anything with respect to taking pyruvate and putting it through the citric acid cycle, we have to get pyruvate into the mitochondria from the cytosol. That occurs via something called a proton pyruvate symport protein. Okay, so we get our little pyruvate into the mitochondria via this little symport protein that lies in the mitochondrial membrane. Okay, so now let's start again with these 5 reactions that are catalyzed by this gigantic pyruvate dehydrogenase multi-enzyme complex. Skip the slide. Okay, so what does this complex look like? Well, it depends on the organism, right? But for the majority of organisms, it consists of 3 separate enzyme subunits, okay? The first one is called pyruvate dehydrogenase, and I'm going to abbreviate these because I don't like to say their names, period. So pyruvate dehydrogenase, we call this E1. The second enzyme that it contains is dihydrolipoamide transacetylase. Again, there's a reason why I don't like to call these by their name. We call this guy E2. And the third one is called dihydrolipoamide dehydrogenase. We call this guy E3. Now, the overall magnitude of this complex depends on the organism that you're in, right? In eukaryotes, this ends up being the largest multi-enzyme complex in the entire organism, right? So this becomes gigantic when we move into eukaryotes. Overall, though, they have very similar type of architectures. So eukaryotes, this is the largest multi-enzyme complex. Okay, so in eukaryotes, we'll stick with that one. I won't talk about E. coli. This multi-enzyme complex, first off, contains 60 separate E2 subunits, okay? So number one, the way it's built is essentially a cube-like structure of 60 E2 subunits. That's what forms the core for this enzyme. Surrounding this E2 core, we get 60 E1 subunits. So again, we have this core of E2. It is surrounded by 60 E1 subunits. And then in that 60 E1 subunits are speckled 12 E3 subunits. Okay, so the core is E2. It is surrounded by 60 E1 and 12 E3s. Okay, very large. Long term, with respect to the size of this ends up being over 10,000 kilodaltons, which is gigantic. Okay, so now, what are the various types, beginnings of the citric acid cycle? Okay, so in this citric acid cycle, long term, we are going to take our little pyruvates that we got from glycolysis, right? So pyruvate is a nice 3-carbon sugar, and we are going to completely oxidize all of those carbons to CO2, the fully oxidized version of carbon. Okay, so those 3 carbons of pyruvate are going to long-term turn into 3 CO2 molecules. Okay, so again, we're going to oxidize these guys. In the process, we're going to produce reducing equivalents in the form of both NADH as well as FADH2, and that's essentially what's going to come out of the citric acid cycle. Okay, before we get into the citric acid cycle, though, we needed to take our pyruvate molecules and turn them into acetyl-CoA. So acetyl-CoA is essentially the way that we start the citric acid cycle. So before we can get pyruvate into the citric acid cycle, it has to be turned into acetyl-CoA. One thing to note about acetyl-CoA, so number one, this CoA is just a coenzyme. I gave you this structure when we first started talking about the citric acid cycle last week. The acetyl group, though, is just a 2-carbon derivative. Okay, so we essentially, in turning pyruvate into the acetyl group on acetyl-CoA, will decarboxylate one of the carbons off of the pyruvate. Because again, when we enter this, we only have 2 carbons left over. So the enzyme complex that catalyzes this reaction, this taking pyruvate and turning it into acetyl-CoA, is something called pyruvate dehydrogenase. Okay, and so I started talking about pyruvate dehydrogenase last time. So with respect to pyruvate dehydrogenase, it is this very, very, very large multi-enzyme complex, right? It consists of a number of different subunits that essentially have 3 separate subunits overall. So the 3 subunits that we deal with in pyruvate dehydrogenase are something called E1. So E1 is termed pyruvate dehydrogenase, so that's one of the subunits. The second subunit is called E2, or this dihydrolipoamide transacetylase. And I'm not going to ever say that again, so just note that's what I'm talking about when I say E2. And then the third subunit, we abbreviate E3, and this is dihydrolipoamide dehydrogenase. Okay, so again, this entire complex consists of a number of different subunits of this E1, E2, and E3. Okay, depending on the organism is going to depend on how many individual subunits that are there. But for the most part, regardless of the organism, the core of this pyruvate dehydrogenase complex consists of these E2 subunits that are surrounded by E1 and E3 subunits. Okay, so in this multi-subunit or multi-enzyme complex, it is going to catalyze 5 separate reactions. So 5 reactions are going to be catalyzed on this large multi-enzyme complex. And in each of those subunits, there are going to be 5 separate cofactors. So 5 separate cofactors are required. So a couple of these cofactors we have talked about before, and we are semi-familiar with at this point in time. So for example, one of the cofactors is just our normal little NAD+, right? Nicotinamide adenine dinucleotide. We have seen this guy before, okay? Another cofactor that we have seen before is this guy, this thiamine pyrophosphate, TPP. Where have we seen this before? So TPP tends to play a role in decarboxylation reactions, and that's what it's going to do here. So in general, we'll see TPP anytime we have a decarboxylation, and specifically with TPP and alpha keto decarboxylation reaction. Okay, so we have to use this guy anytime we have this alpha keto decarboxylation because the transition state is too unstable in the alpha ketone. Okay, so we've also seen this already in the turning acetaldehyde or turning pyruvate into ethanol. That is where TPP came in. There was an enzyme that catalyzed that decarboxylation. It also used TPP as a cofactor. Okay, so a couple other things that we haven't seen before. So I did show you coenzyme A, and I showed you the structure of coenzyme A. There is going to be another cofactor called lipoic acid, and I will show you the structure of that, as well as another cofactor called flavin adenine dinucleotide, and we will talk about that, okay? So a couple of things right now, because we haven't really spoke of this flavin adenine dinucleotide yet, so I'm going to spend a little time dealing with this. So flavin adenine dinucleotide, or FAD, is very similar to NAD, right? It just doesn't have a nicotinamide group. Instead, what it has is a flavin. Okay, so flavins are nice and interesting little creatures. They essentially are, at least the functional group of a flavin is this nice three-membered cyclic ring type structure, which is called as a group, and I have to look this one up as usual. This is called an isoalloxazine group. Okay, so again, this is a nice delocalized three-member cyclic ring structure. Okay, so a couple things to note about FAD, these flavins versus NAD, is the fact that flavins tend to be either non-covalently or covalently associated very strongly with the enzyme that they are in, okay? So flavins tend to stick to the enzyme, either if that is via covalent attachment or a very strong non-covalent attachment. That is not the same with respect to NAD, right? So NAD tends to enter the active site of the enzyme, the reaction tends to occur, and then it tends to diffuse away and go wherever it needs to go. Flavins do not work like that. They are going to be either covalently attached or they're going to be very intimately associated with the enzyme. They are not going to diffuse out of the active site, okay? Another important thing to note about FAD versus NAD is the fact that for NAD, if we look at the overall reduction, so we have NAD plus 2 electrons plus a proton forming our NADH, our nice reduced cofactor. For NADs, these guys can only ever undergo 2 electron oxidation reductions, right? So with respect to NAD, if it's going to transfer electrons, it has to transfer 2 at a time. If it's going to accept electrons, it also has to accept 2 at a time. So this can only come in the form of these hydrides. So flavins are different. So flavins can undergo both 1 electron and 2 electron reductions, okay? So the reason why is because they have a nice stable, what we call semiquinone radical intermediate. So you can transfer 1 electron and it can be nice and stable and be fine, or you can transfer 2 electrons and get the fully reduced FAD plus, FADH2, pardon me. Okay, so again, flavins, you can have both 1 and 2 electron transfers. That's going to become important with respect to the electron transport chain. And then for NAD, you can only have 2 electron transfers. And that's, again, going to be important with respect to the electron transport chain. Okay, so let's get back to this overall reaction with respect to this pyruvate dehydrogenase complex, right? So now we have our 5 different cofactors, we have our 3 subunits. So now let's go through these 5 reactions that this multi-enzyme complex is going to catalyze. Okay, so the first reaction is going to be catalyzed by E1. So just as a reminder, E1 stood for just pyruvate dehydrogenase, okay? So in E1, which is going to be along the outside core of this multi-enzyme complex, we're going to have our pyruvate, which is a substrate here, entering with our little pyruvate, and it is going to interact with our TPP cofactor that is bound covalently to this E1 subunit, okay? So a couple things to note about this TPP cofactor that I don't know that you covered when we talked about it in glycolysis. Okay, so this TPP cofactor, the major functional group here, or the major redox active group, or just active group period is something called a thiazolium ring. So this guy, this overall structure, is called a thiazolium ring. And there's a reason why this has to be used in these alpha keto decarboxylations. Okay, this thing is going to act as a nice electron sink. Okay, what we're going to get in the formation of any type of decarboxylation reaction here is the production of a carbanion. Right, this carbanion is going to have a negative charge on carbons, which carbons don't like negative charge on them. So TPP is going to help stabilize that via resonance. Okay, so that's the role that TPP plays here. So a couple other things to note. So with respect to this TPP cofactor, this thiazolium ring, in the neutral normal version, this carbon right here is protonated, right? So neutral normal version, this carbon is protonated. In the active version, though, it is deprotonated. So this carbon, if it had a hydrogen there, is very acidic, right? So it will deprotonate. And when it does so, it forms this active version of TPP. Okay? So note that if it's deprotonated, we have, again, this formation of this carbanion. There's a lone pair of electrons on this carbon, giving it an overall negative charge. We have right next to it a positive charge on the nitrogen. Okay? So this is something that is formed. It's called a dipolar ion, where you have two charges right next to each other, or more importantly, in organic chemistry terms, this is called an ylid. Okay? Anything that has a positive that is right next to a negative is called an ylid, spelled Y-L-I-D. Okay, so again, that carbanion is stabilized by the resonance that is present in TPP. So what's going to happen in this first reaction? So TPP is going to be deprotonated, right? So it's going to have its little negative charged ylid form. It is going to nucleophilically attack the ketone that is on pyruvate, okay? So in the process of that nucleophilic attack, we're going to lose the carboxylate, the CO2 that is over here, and we're going to form this stable intermediate. This is a covalently bound intermediate to the cofactor. This is called hydroxyethyl TPP, okay? So again, note that with respect to pyruvate, we had 1, 2, 3 carbons. We have lost one of them in this decarboxylation, and we are now left with just 1, 2 overall. Okay, so also note that in this process, we do have this carbanion on this hydroxyethyl intermediate. That is a negative charge on the carbon. Again, that's fine, okay? It is stable because this TPP acts as this electron sink. So there is a resonance structure that you can draw for this that helps overall stabilize this carbanion. So it forms. That's, again, the whole purpose of this. Okay, so that is reaction 1. This is catalyzed on the E1 subunit of the pyruvate dehydrogenase. We have pyruvate comes in, it reacts with TPP, and we get the formation of this hydroxyethyl TPP intermediate. Okay, so now reaction 2, because again, there are 5 of these that we're going to talk about. So what's going to happen in reaction 2? So reaction 2, again, stays on E1. Okay, so we're still in E1. But what's going to happen is we're going to have the movement of another cofactor into the active site of E1. And that second cofactor that's going to come in is this lipoamide. Okay, so lipoamide, let me just go ahead and write that out. So lipoamide is a cofactor that consists of 2 different parts. Okay, the first part is something called lipoic acid. Okay, so lipoic acid is this large hydrocarbon chain that terminates in this nice little heterocyclic ring system. It's important to note that this heterocyclic ring system contains a disulfide bond right at the end of it. this is covalently attached to a lysine that is present on the E2 subunit. Okay, so we have a covalent attachment of this lipoic acid to a lysine, and this lysine comes from E2. Okay, so what type of attachment or what type of bond is formed between this lipoic acid and our lysine here? Peptide bond. What is another word for a peptide bond? An amide, right? So this is why we call this lipoamide. Probably pronouncing that incorrectly, but that's why we call this lipoamide. Okay, so a couple of things about this cofactor, okay? So in this form, with this disulfide bond at the end, this is the oxidized form of this cofactor, okay? So up here, this guy is oxidized, and it forms this disulfide ring system. This can be reversibly oxidized and reduced. Okay, so if we reduce this disulfide, we get the formation of this dithiol. Okay, so again, we can reversibly oxidize and reduce this with electrons and oxidizing reagents. So in the oxidized version, we call this lipoamide. In the reduced version, we call this dihydrolipoamide. Okay, so reduced, you get the two dithiols. Oxidized, you get the disulfide. One other important little thing to note is this lipoic acid attached to a lysine, right, forming this long thing, forms overall a tether that is about 14 angstroms in length. Okay, so this is going to come in handy, or knowing this is going to come in handy with respect to the overall mechanism of this PDH complex. Just note that this lipoamide is very long, and you can think of it as a nice little tether. Okay, so again, we haven't even talked about reaction 2. We've only talked about this lipoamide. So what happens with respect to reaction 2? So our little lipoamide and its oxidized version is going to move into the active site of our E1 complex, and it is going to be attacked by the carbanion of the hydroxyethyl TPP. Okay, so we have nucleophilic attack from our little carbanion. In the process, that hydroxyethyl group, which is now an alcohol, is going to be oxidized to a ketone. Okay, so we have an alcohol which is going to be oxidized to a ketone, and the electrons that were present in the alcohol are going to be used to reduce that lipoamide. Okay, so we get reduction of the lipoamide to the dihydrolipoamide. We also had something that was an alcohol that is now a ketone, and this is our acetyl group that is going to move on with respect to our process. Okay, so these are the two carbons that are going to form our acetyl-CoA. Okay, so at the same time, by doing this reaction, we have re-yielded our nice ylid form of TPP that can go back on E1 and undergo further reaction. Okay, so now we have transferred our little acetyl group to our dihydrolipoamide. We're going to move on with that with the remainder of the reactions. Okay, so now it was an E1, so now it's going to move to E2. Okay, so in E2, we have brought our little acetyl group via our lipoamide cofactor. In E2, what's going to happen is it is going to react with acetyl-CoA. Right, so coenzyme A, pardon me. So it's going to react with coenzyme A. What we're going to get is something called a transesterification reaction. Right, so essentially all we're doing is transferring this acetyl group or acetyl group to coenzyme A to form our acetyl-CoA. So a couple things to note about this. Even in this acetyl dihydrolipoamide cofactor, we had a very high energy bond formed, right? So this guy right here this sulfur to carbon to the carbonyl. What is this called? If instead of it was S, if it was oxygen instead, what would that be? Oxygen to a carbonyl would be an ester. So this is called a thioester. Okay, so thioesters are very, very high energy. So their hydrolysis long term can be utilized in general to produce things like ATP, etc. So note that we formed a very high energy bond in the production of this acetyl dihydrolipoamide, and that high energy bond is conserved whenever this is transferred to coenzyme A. Okay, we still have this nice high energy thioester bond. Okay, so we've transferred it. We're on E2. That is why E2 is called the dihydrolipoamide transacetylase, because it essentially just transfers the acetyl group from the dihydrolipoamide to coenzyme A. So now we have our acetyl-CoA, right? Acetyl-CoA is going to move off to the citric acid cycle. That's how we're going to start our citric acid cycle. Right, so that should be it. 3 reactions, right? Completely done. Moving on. Not really, right? How many more reactions do we have to talk about? We have 3 reactions, but how many reactions does this actually catalyze? 5, right? So acetyl-CoA, that's great. It's going to move off and do its own thing. What do we need to do, though, to get this back to the state that it was in when we originally found it, right? Because that's the purpose of a catalyst. So right now, what is the oxidation state of our little lipoamide? Is it oxidized or reduced? It's reduced, right? How did we find it? Oxidized, okay? So the next 2 reactions are essentially going to reoxidize our little lipoamide cofactor, right? So that we can get it back to the state that we found it in so that we can do more reactions with it. Okay, so where's that going to occur? What are we going to do with respect to this guy? So to reoxidize our lipoamide cofactor, we are now going to move to subunit E3, which is the dihydrolipoamide dehydrogenase. Okay? As a note, I'm just going to write this dehydrogenase. A dehydrogenase almost always guarantees production of something like NADH, reducing equivalents, okay? Anytime we use the word dehydrogenase, that should tell you that probably NADH has been produced in that reaction, okay? So again, E3 has dehydrogenase as part of its name. So if we need to reoxidize our hydrolipoamide or dihydrolipoamide, then it's probably going to be at the expense of giving those electrons to NAD to make NADH. So how is that actually going to occur? Okay, so now we're going to take a little lipoamide, we're going to move it to E3. In E3, what we have is 2 cysteines that are very close together. Okay, so in the active form of this, these guys form a disulfide bond. And again, disulfides are the oxidized versions of these 2 cysteines. So this is the thiol from one, thiol from the other, and again, it is oxidized, so it has this little disulfide bond. So the dihydrolipoamide is going to meander in to the active site. The electrons are going to reduce our little disulfide to produce our nice little cysteines, our dithiols. And we are left, at this point in time, with our reoxidized lipoamide, okay? So this is going to go off and be ready for more reactions. So again, this is reaction 4. All we're doing is essentially exchanging a disulfide bond between the dihydrolipoamide and the disulfide between the 2 cysteines. Okay, so good. We have our Lipoamide, it's reoxidized, so it's back to its active form. What else do we still have to do to make sure that we get this back to its original state? What happens to these guys? We need to reoxidize them, right? Right now, we have this disulfide between the two cysteines. It is reduced. To get this back to its normal, original form, we need to reoxidize them. And so that's where the NAD plus comes in. Okay, so that's the last step in this process. We have our reduced dithiol between the two cysteines. So this is a reduced form. The electrons that are in this reduced dithiol are going to be transferred to NAD plus to form NADH, which is our nice little reducing equivalent. In the process, we're going to reoxidize this dithiol to this disulfide. And we are ready for our next cycle with respect to the enzyme. Okay, so one thing to note about the overall mechanism of this enzyme, if we zoom in on the active site of E3, this dehydrogenase, we'll find that our cysteines are right here. This is the disulfide that forms between these guys. We'll also find that this FAD, this flavin adenine dinucleotide, this guy is covalently attached to E3, right? So it lies essentially in between where this disulfide is, between the two cysteines, and where the NAD plus comes in to take the electrons from the two cysteines, okay? So you would think at some point in time that if the electrons are going to be coming from these guys, right, that they probably need to pass through FAD to get to NAD plus, okay? So there's a little bit of an issue with that, and this goes back to the concept of reduction potentials. So with respect to FAD, the reduction potential of FAD, where we have 2 protons, 2 electrons forming our FADH2, which is what would happen if we actually reduced FAD plus, or FAD. This has a potential of approximately 0 volts. Okay? So this is the reduction potential for FAD. For NAD plus 1 proton plus 2 electrons going to NADH, this has a reduction potential of about minus 0.315 volts. Okay, so is it capable, if FAD is reduced, would FADH2 be capable of transferring electrons to NAD plus, given those reduction potentials? So no, it would not, right? It has too high of a reduction potential. So FAD is a very strong oxidant. It would not be able to, or FADH2 would not be able to reduce NAD plus, okay? So there's a little bit of an issue here in thinking that the electrons actually go through FAD. So most scientists believe that the role of FAD here is not to be formally reduced, but to essentially act as an electron conduit, right? Kind of like a nice little wire by which the electrons will move from the disulfide into the NAD plus without ever formally reducing the FAD. Okay, so FAD is believed to act as an electron conduit in this reaction. And there's really no proof that it ever gets formally reduced. So just imagine it's an electron conduit or a nice little molecular wire, if you will. Okay, so that is yes. Sorry, what? Sorry, again. It's never formally reduced, no. And you can see that spectroscopically. You never actually see the formation of the reduced FADH. Okay, so that is the reaction 5 catalyzed by the pyruvate dehydrogenase complex. Okay, so we returned everything to its original state. The electrons that came from the pyruvate are now on NADH, and we've also produced a nice little CO2 molecule. Okay, so a couple of things to note about how this overall multi-enzyme complex works, right? So notice that lipoamide formed the majority of things that happened with respect to this overall complex, right? So if we remember back, lipoamide was on E2, right? It somehow managed to swing in to E1, right, where it picked up the hydroxyethyl from the hydroxyethyl TPP. It transported that to E2, where it was at. So we were at E1 originally. It then transported that to E2, right? So that was the next step in this overall reaction, where we transferred the acetyl group to coenzyme A. It then itself, because it was reduced, moved into E3, where it was reoxidized and everything was returned to normal, okay? So how does this do this? Because again, imagine that you have this very, very, very large multi-enzyme complex. So the length of the lipoamide cofactor is very important in doing this, right? So it's essentially acting like this little 14 angstrom tether that's moving from one active site on one subunit to another active site on another subunit, and so on and so forth, okay? Another important thing to note about this is that this lysine, or this guy is covalently attached to E2, is covalently attached on a very flexible portion of the enzyme. So it's about 140 angstrom portion of E2 that consists primarily of alanine and prolines, right? So it gives us this nice little flexible movement so that the tether can move between E1 and E2 and E3 and catalyze its various reactions. Oh gosh, did that really take that long? Okay, so that's it. So now we're into the citric acid cycle, right? We formed acetyl-CoA. Now we can do the citric acid cycle, so that's great. So let's test a couple of concepts here. If you guys remember Socrative, go ahead and get your answer in. so what is the correct answer here? B. All right, so pyruvate is oxidized. So just note, anytime we have a decarboxylation, I probably said this before, that usually is accompanied with an oxidation. Okay, so anytime you take off a CO2, you're going to oxidize the thing in the process. So pyruvate is oxidized to acetyl-CoA. All right, what about this one? I want to know which of these statements is false. Yeah, I'm opening. Okay, so go and get your answers in if you haven't yet. End this. Okay, so what is the correct answer for this one? It was C. All right, so everything else is correct. Where does this pyruvate dehydrogenase complex, where does it take place? Where does it do its activities? In the mitochondria, right? So remember, we had to take pyruvate, we had to export it into the mitochondria from the cytosol, and so everything occurs in the mitochondria. The trick. Yep. This is inside, not in the membrane. There's going to be one enzyme that's actually part of the membrane, but the majority of it is taking place inside the mitochondria. And the cytosol of the mitochondria. I guess it's not the right thing to say. But anyways, okay, yeah, so moving on. Let's talk about now our citric acid cycle, right? So we have our acetyl-CoA. We have produced our acetyl-CoA via our nice little pyruvate dehydrogenase complex, okay? So again, we have a nice little high-energy thioester bond that is present in our acetyl-CoA molecule, okay? So we're going to enter our citric acid cycle, and in the citric acid cycle, there are going to be 8 separate reactions, okay? So those 8 reactions are going to consist of condensations, isomerizations, decarboxylations, oxidation reductions, as well as phosphorylations. So lots of different things are going to happen. Overall, though, the big purpose of this is we're going to take the acetyl group on our acetyl-CoA. So we have our nice little 2-carbon fragment. There are 6 electrons that can be derived from the methyl of the acetyl group. So we're going to take 6 electrons from this. There are 2 electrons that can be derived from the carbonyl of the acetyl group. And we're going to utilize those to produce overall 2 CO2s. We're going to completely oxidize them. That gives us 8 electrons, or 4 pairs of electrons. That's going to be used to produce 3 NADHs. So 6 of those electrons are going to go into the formation of NADH, as well as 1 FADH2. Okay? We are also, in the process, going to form a GTP, which is synonymous with ATP, and we'll talk about that when we get to the formation of our GTP. Okay, so 8 separate reactions, lots of different chemistry that takes place in each of them. So what is the first reaction? So the first reaction is actually going to be a condensation reaction. We're going to take our acetyl-CoA, it's going to enter, and it's going to react at an enzyme called citrate synthase. Okay? At citrate synthase, at this enzyme, we're going to condense the acetyl portion of our acetyl-CoA with the 4-carbon metabolite oxaloacetate. Okay? So we have 4 carbons on oxaloacetate. We have a 2-carbon on the acetyl group. They're going to condense and form a 6-carbon metabolite called citrate. Okay? Okay, so there's a couple of things I want to note about this mechanism before I actually go over the mechanism. So citrate synthase operates via something called an ordered sequential mechanism. And I didn't really talk about this when we dealt with enzyme kinetics, but the idea behind an ordered sequential mechanism is that for this enzyme, we have 2 separate substrates, right? We have oxaloacetate and acetyl-CoA. So 2 different substrates. With respect to ordered sequential mechanism, there is going to be a specific order in which each substrate must bind, okay? So for this mechanism, oxaloacetate has to bind first. And it binds to a form or a conformation of the enzyme called the open form. Okay, so there are 2 different conformations. Oxaloacetate binds to the open form. When it binds, the enzyme itself undergoes a conformational rearrangement to form what is called the closed form. So you can see there's a rotation about a small little subunit, and it forms this closed form. Again, that happens upon oxaloacetate binding to the enzyme. At that point in time, that closed form will open up the binding site for acetyl-CoA. Okay, so for the actual binding site with respect to acetyl-CoA, it is not opened until after oxaloacetate is bound. The enzyme undergoes that conformational change to the closed form. Then acetyl-CoA will be able to bind at that point in time. So this is what we mean by ordered sequential. One comes first, changes the conformation so that the other can bind. Okay, so what is the actual mechanism by which we have this nice little condensation reaction between our acetyl group and oxaloacetate? So just as a reminder, that acetyl-CoA had a nice high-energy thioester. And we're going to see essentially the waste of that energy in this reaction. We're going to talk about why We see this waste when we get to the end of the citric acid cycle, okay? So what's going to happen? We have oxaloacetate. It comes in and it binds, closes the enzyme, which opens up the binding site for acetyl-CoA. So the first step in this mechanism is just general acid-base catalysis, okay? So we have an aspartate that's going to act as a general base. It's going to abstract a proton from our acetyl group, and we're going to have a histidine that's going to act as a general acid. It's going to donate a proton to our acetyl group, forming the enol version of this compound, okay? So with respect to an enol, what is an enol? It is a alkene what? Alkene alcohol, right? That's why it's the ol, it's an alcohol. So we have an alkene alcohol, pardon me. That's what an enol is. That enol is nice and nucleophilic. It is going to attack the carbonyl of our oxaloacetate, okay? And we're going to get the formation of this actual covalent intermediate citryl-CoA, okay? So we have general acid-base catalysis. This guy was a general acid. We get the formation of our enol, which is going to nucleophilically attack the carbonyl on our oxaloacetate. We get the formation of our covalent citryl-CoA intermediate. This is then going to hydrolyze, and it's going to hydrolyze that thioester bond. And so we're going to lose, in this third step, our coenzyme A, okay? So we get this nice hydrolysis of the thioester, and we get the formation of citrate, which is our condensed 6-carbon metabolite, okay? So again, this was a really high energy bond. The delta G for this reaction ends up being a minus 31 kilojoules per mole, okay? And that is not put in to do anything, right? We don't synthesize ATP from that. We don't produce any type of reducing equivalents or anything like that, okay? So right now, in this first step, we have just thrown away 31 kilojoules per mole in our nice high energy thioester, okay? I will show you the chemical logic behind this one when we get to the end of the cycle, if we ever do. Yeah. The major driving force is the hydrolysis of that. Oh, no. We'll talk about regulation overall. There's a couple different steps that have very exergonic delta Gs. Okay, all right, so that's one, citrate synthase. We take acetyl-CoA, we take oxaloacetate, and we get citrate. Reaction 2 is going to be catalyzed at this beautiful little enzyme called aconitase, okay? So what's going to happen at aconitase is we are going to take citrate, which is an alcohol, and we are going to isomerize it, right, to form isocitrate. Okay, isomerization reaction. All we're really doing is changing the position of that alcohol group. So how do we do that? Well, the first thing we do is we dehydrate our little citrate. So we have citrate here. If I undergo dehydration, what am I going to form? I'm losing a water, but what do I usually form in the process? A double bond, right? So this is a nice classic elimination reaction. We eliminate water and form a double bond. That's the first step of the aconitase reaction. In the second step, I add water to the double bond. What is that called? It's not elimination, but if I add water, if I hydrate the double bond, what is that called? Just addition, right? So we undergo an elimination followed by an addition, where essentially we just add water again. So we eliminate water and then we add water. Long term though, what does that do? We take the OH, which was here, and we move it to this position here, okay? That may seem kind of silly, right? But there is chemical logic to this overall reaction. So we take citrate. What type of alcohol is citrate? Is it a primary, a secondary, a tertiary alcohol? It's a tertiary alcohol, right? So the carbon that is directly attached to our alcohol group is attached to 3 other carbons, right? So this is a tertiary alcohol. When we turn this into isocitrate, what type of alcohol do we have now? This is a carbon that is attached to 2 other carbons. So this is a secondary carbon, okay? In the next step, of this reaction, so reaction 3, we are going to want to oxidize that alcohol to a ketone. If we oxidize the alcohol to a ketone at a tertiary position, what am I going to need to break in the process? Carbon-carbon bond, right? Which costs lots of energy and is fairly unfavorable. If I instead try to oxidize an alcohol at a secondary position, what do I have to break? Just a carbon-hydrogen bond, right? Much easier to do, okay? So that is the chemical logic behind the aconitase reaction. We take something that is a tertiary alcohol, convert it to a secondary alcohol for our next step in the reaction, which is oxidation of that alcohol, okay? Okay, just as a little note with respect to pub quiz type chemistry or anything like that, if you're ever participating, aconitase, as an enzyme, does have a cofactor in it, right? This is not a redox reaction, though, so I want to make note of that. This is just a simple elimination and then addition reaction, just a simple isomerization. It has a cofactor, though, that usually participates in redox-type reactions. So specifically, it has a 4-iron 4-sulfur cluster as a cofactor. Now, the only reason why I mention that is because this is the only enzyme in either glycolysis or the citric acid cycle that contains this type of 4-iron 4-sulfur cluster, okay? So this is the only enzyme in either glycolysis or citric acid cycle that has a cofactor like this. And usually these cofactors take place or are found in enzymes that undergo redox types reactions, okay? So we will find more 4-iron 4-sulfur clusters in the electron transfer chain. But for right now, this is the only one that's present in glycolysis or the citric acid cycle. Why this is interesting is because aconitase actually moonlights in another role in the body. So aconitase acts as an iron sensor and essentially is capable, because of various conformational rearrangements, is capable of turning on the proteins that are responsible for both sequestering iron as well as transporting iron in the blood. Okay, so it has two roles. It plays a role in the citric acid cycle, but it also acts as an iron sensor. Okay, so aconitase, right? So now, reaction 3. Reaction 3 is we're going to start doing some real chemistry, okay? So we have converted citrate into isocitrate, which is a nice secondary alcohol at this point in time. So in reaction 3, which is going to be catalyzed on an enzyme called isocitrate dehydrogenase, we're going to oxidize that alcohol to a ketone, okay? So that's the first step. Again, dehydrogenases, what do they produce usually? NADH, right? So they usually oxidize something and produce reduced cofactor, NADH, in the process. So first step is that oxidation. We're going to form an intermediate called oxalosuccinate, okay? Now this is a beta keto acid, okay? So I said that alpha keto acids had to use TPP to be carboxylated or decarboxylated. Beta keto acids, though, don't need TPP. So the second reaction in this is going to be a decarboxylation reaction where we lose CO2, remove this little carboxylate group. But because this is a beta keto acid, we don't actually need TPP to do that. Instead, all that we require is divalent metal cations. Okay, so this could be manganese 2+. This could also be magnesium 2+. The divalent metal cation, its role is just to stabilize the negative charge that develops in the transition state. And then after we form our oxalosuccinate, we get decarboxylation. We are then, long-term, going to form alpha ketoglutarate. Okay, so isocitrate had 6 carbons. This is our first decarboxylation as well as oxidation. So now we are left with 5 carbons in alpha ketoglutarate. This is also the first decarboxylation, oxidation in the cycle. So this will be another key important regulatory point with respect to citric acid, because again, this is kind of the first I guess, money in on the overall reaction. Okay, so we have generate alpha ketoglutarate. Alpha ketoglutarate is an alpha keto acid. So the next step in this, so this is alpha keto acid. The next step in this is going to be another decarboxylation as well as oxidation. We're going to take alpha ketoglutarate and we are going to form succinyl-CoA. Okay, this is going to be catalyzed on something called alpha ketoglutarate dehydrogenase, which is great because it is essentially the exact same thing that we've already talked about, pyruvate dehydrogenase. Okay, so the overall multi-enzyme complex that takes alpha ketoglutarate and converts it to succinyl-CoA is a multi-enzyme complex that resembles overall the pyruvate dehydrogenase that we talked about at the very beginning, which took pyruvate and turned it into acetyl-CoA. Okay, it has the exact same 3 subunits. Okay, so it has an E1, an E2, and an E3. It's this exact same multi-subunit gigantic complex. Okay, it's got the exact same cofactors. The only difference is instead of pyruvate, it starts with alpha ketoglutarate, and instead of acetyl-CoA, it forms succinyl-CoA. Okay, but again, we're going to see a decarboxylation as well as an oxidation, which is going to produce NADH. So with alpha ketoglutarate, we had 5 carbons. And it undergoes this decarboxylation. We now are left with 4 carbons in the form of succinyl-CoA. So how do we start this citric acid cycle? We brought in pyruvate from somewhere else, but what else did we condense pyruvate with? Oxaloacetate. Okay? How many carbons does oxaloacetate have? 4. Okay? So we're at the point where we're back to 4 carbons. We have decarboxylated the 2 carbons off. Okay? So now the last steps in this overall citric acid cycle is taking succinyl-CoA and getting it back to oxaloacetate. Okay? Also, really important point to note, which I have not noted thus far, and I should have, and I apologize for that, is the carbons that were from pyruvate are up here. And I want my highlighter. Right here and right here. Okay? These carbons that we've been taking off in the citric acid cycle are not the carbons that we put on when we put pyruvate in. Okay? So the pyruvate carbons stay here. They will come off in subsequent cycles. And if you wanted to track those carbons and when they would actually come off, that would probably be very intelligent. Okay, and one more thing, right? We get this coenzyme A attached to our little succinyl group, right? Our succinyl molecule, whatever. So we also get the production of a high energy thioester bond. The first thioester that we made, we wasted, right? We just let it go. This thioester, though, we're going to use, right? Because again, this is very high energy, so we can utilize the energy of its hydrolysis to produce something. What do you think we're going to produce? Not NADH. What else do we produce? So we have nice high energy bonds, ADP or ATP, right? So that's what's going to come next. We have our succinyl-CoA. In reaction 5, we are going to produce something called succinate, right? In the process, we are going to hydrolyze off our coenzyme A. I'm very tired. I'm not having my thing that I think I have going. So we're going to hydrolyze coenzyme A. We're going to use that energy to phosphorylate either GDP or ADP. Okay, so this depends on the actual tissue that you're present in. Okay, so this could be either ADP to ATP or GDP to GTP. How does it depend on the tissue that you're in? Well, in oxidative tissues, so oxidative tissues are going to be things like muscle and heart, etc. The product of this is ATP. So ADP is what's going to be used in substrate level phosphorylation. On the other hand, in more anabolic tissues, So anabolic tissues are things like the liver. GTP is going to be formed instead. Okay, so what does this reaction do? So again, the first step in this is we're going to take inorganic phosphate, nice little PI. It is going to act as a nucleophile. We're going to see a nucleophilic substitution reaction. So it is going to attack the carbonyl of our thioester. We get this nice little nucleophilic attack. Coenzyme A is going to act as a leaving group in this, and we are going to generate something called succinyl phosphate. Okay, so we have lost our little coenzyme A, but we have attached a phosphate group to our little succinyl molecule. Okay, all right, so now in the second step, we have a histidine residue that is directly attached to the enzyme. So this is covalently bound to the enzyme. It is going to, again, act as a nucleophile. In this reaction, the histidine is going to attack the phosphoryl group, and succinate is going to act as a leaving group here. So we generate what is called a phosphohistidine residue, and we have succinate, which is part of our citric acid cycle product here. So this is going to move off onto the next step. Now to utilize this little phosphohistidine to phosphorylate GDP or ADP, the GDP or ADP is going to come in, and essentially all we're going to see here is this phosphoryl transfer from histidine to either GDP or ADP to generate GTP or ATP. And that gives you a nice little formation of ATP from the hydrolysis of that thioester of the succinyl-CoA. So that's reaction 6, or that's reaction 5. There are only 3 more, and it's only 2 more slides, right? Because now we've got succinate. It's a 4-carbon sugar. All we need to do is get it back to oxaloacetate, which is, again, 4 carbons, right? We're done with generating anything. All we have to do is isomerize some stuff and get it back to where we started at. So the final reactions, the citric acid cycle, is going to be reaction 6 and then 7 and 8. So we're going to start by taking succinate, which again, 4 carbons, and we are going to oxidize it, right? So this is a little bit different. We're not eliminating things, we're instead oxidizing it. If we oxidize it, there is no alcohol to oxidize, okay? There's nothing that we're going to oxidize to a ketone or an aldehyde. So instead, what we're going to do is oxidize a carbon-carbon bond, okay? In the process, we're going to get a carbon-carbon double bond formed. Now, the big point of this, or what you need to take away from this guy, is the fact that carbon-carbon bond oxidation is much harder than carbon-oxygen bond oxidation, okay? So for carbon-oxygen, anytime we've taken some sort of alcohol and oxidized it to either an aldehyde or a ketone, what have we used to do that? NAD+, right? So every time we've taken any type of alcohol and oxidize it to a ketone or an aldehyde, we've utilized NAD+ to do that. That is the oxidant that we use. A carbon-carbon bond, though, is going to be harder to oxidize, so we need a better oxidant to do that. Because of that, we have to use the better oxidant, which is FAD, okay? So anytime you see carbon-oxygen bond oxidation, it's always NAD+. Anytime you see carbon-carbon bond oxidation, the cofactor has to be FAD, okay? So we get fumarate as our product of this carbon-carbon bond oxidation, as well as reduced FADH. Now this reaction is catalyzed on an enzyme complex called succinate dehydrogenase, okay? Now, only thing to note about this, we're going to come back to succinate dehydrogenase. This is the only membrane-bound enzyme of the citric acid cycle. Okay? And what this thing is essentially going to do is directly feed electrons into the electron transport chain in the form of complex 2. And again, we'll come back to that to discuss exactly how that happens in the next chapter. Okay, so succinate gets oxidized. We form fumarate, which has a carbon-carbon double bond. So now the final reactions, reactions 7 and 8, we're going to take fumarate and we are going to hydrate it, right? So another addition reaction where we're going to add water across the double bond to form our hydrated product, which is malate. The enzyme that catalyzes this is fumarase. And again, this is just the addition of water. I don't know why I can't think. Addition of H2O across the double bond. And then finally, to take malate, which is an alcohol, and to get it back to oxaloacetate, which is a ketone, we need to take our nice little alcohol. What do we need to do to it? Oxidize it, right? So the last reaction is called malate dehydrogenase, which again, dehydrogenase tells you you're going to use NAD plus as an oxidant. You're going to produce NADH in the process. And we're going to take our little alcohol, we're going to use our nice little NAD plus oxidant, and we're going to oxidize it to a ketone, which gives us oxaloacetate, and it also gives us the final reducing equivalent of the citric acid cycle. Now, one little last thing, because I have like 5 minutes, 3 minutes left, 4 minutes left. This reaction is incredibly endergonic, right? Reaction H is very, 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 I can't stand this. Okay, delta G is very, very, very large and positive. It ends up being about 29 kilojoules per mole, okay? So this is endergonic, it's unfavorable. How does this ever occur? So what reaction does oxaloacetate immediately get used in this overall cycle? The citrate synthase, right? What was the problem that I mentioned originally about citrate synthase? We hydrolyzed that thioester bond, and all that energy just essentially went to nowhere, right? But now, why do we need that citrate synthase? Because essentially, the minute any little molecule of oxaloacetate gets produced, it's going to be immediately consumed, okay? So coupling citrate synthase to this malate dehydrogenase reaction drags this reaction along, right? The minute this guy gets used in citrate synthase, this thing has to come back to equilibrium, so it's going to push this reaction forward and essentially drags it along to completion. So we couple a very exergonic reaction to a very endergonic reaction to get an overall favorable process. Okay, so that's all I have to say today. Have a nice Thanksgiving.